Welcome to this presentation. In this presentation, let us have an insight of electrophysiology and biopotential reading. And at this end of the session, the students will be able to analyze the human body, electrophysiological parameters and recording of biopotential. And the diagram below shows the fundamental unit of every plant or animal or human, and this is cell, and the cell is the basic living unit of the human body, and the combinations of cells is called tissues. Every organ in the body is made of a combination of many tissues. All cells are same and they contain gelatinous substance made up of composed of water, protein, acids, fats and various minerals. Water is the principal fluid in cell concentration of 70 to 80% used in solvent of all chemicals in order to produce chemical reaction. And next you can see here is the cell membrane. It protects the cell and surrounds it that passes into and out of the cell. Cells cannot be seen by naked eye. It can be viewed only by biological microscope. And what you see here is the nucleus. Here. This is a nucleus. A nucleus controls the structure of the cell, and the cell reproduction process is directed by the nucleus only, and which determines the function of the cell and the structure of the cell. And the cytoplasm is the which carries the work of a cell. Cyto means cell, plasm means formation. It is cell formation. Cytoplasm contains mitochondria and endoplasmic reticulum, and this mitochondria is the power center of the cell, and the metabolism, the combination of catabolism and anabolism is called the metabolism. In this process, the sugars and fats in the food are used up and burned quickly, so energy is released. And now let us see the sources of biomedical signals. Certain types of cells within our body, such as nerve cells or muscle cells, are encapsulated in semi-permeable membrane that permits some substances to pass through the membrane while others are kept out. Surrounding the cells of the body are the body fluids, which are ionic and provide a conductive medium for electric potentials. And the principal ions that involved in the phenomena of producing cell potentials are sodium, potassium and fluoride. So the membrane of the excitable cells readily permits the entry of potassium and chloride ions, but effectively blocks the entry of sodium ions, even though there may be a very high concentration gradients across the cell membrane. This results in the concentration of sodium ions more on outside than the inside membrane. The various ions balance between inside and outside according to the concentration of electric charges. And the membrane potential caused by different concentration of ions is called resting potential. Now the cell is called in the resting state, that is polarized state. Okay, and the cell membrane is excited by flow of ionic current or some form of externally applied energy, and the membrane changes its characteristics and begin to allow some sodium ions to enter. And this movement of sodium ions constitute an ionic current flow that further reduces the barrier. So the net result is an avalanche effect in which sodium ions literally rush into the cell and try to reach a balance outside. At the same time, the potassium ions were in higher concentration inside the cell during the resting state and try to leave the cell but unable to move as rapidly as sodium ions. And as a result, the cell has a slightly positive potential or inside and due to the imbalance of potassium ions, and this potential is called active potential. Usually the action potential is about 20 millivolt. And the positive potential of the cell membrane during excitation is called action potential actually, and the cell that has been excited and that displayed an action potential difference is said to be depolarized, and the process of changing from resting state to action potential is called depolarization. And when the passage of sodium ions is stopped, the cell membrane reversed back to the original condition, and that is called repolarization. Okay, and thus repolarization can be seen in this diagram. So once the rush of sodium ions through the cell membrane has stopped, the ionic currents that lowered the barrier to sodium ions and the membranes reverts back to its original. An active process called sodium pump, which is used to keep the passage of sodium ions from outside and potassium ions inside, it use short time for resting potential to develop again. So sodium ions are quickly transported outside the cell again here and become polarized, and this process is called repolarization. And the typical action potential form, beginning at the resting potential, depolarizing and returning to the resting potential after repolarization, that you can see in this figure. It appears as like a spike of 1 millisecond duration, and it depends on the types of cell producing the potential. For example, it varies from cell to cell. For example, for in nerve and muscle cells, repolarization occurs so rapidly, that is 1 millisecond. And for heart muscle, the repolarization is very much slower. It takes around 150 to 300 millisecond, right? Now let us see the all or nothing law. Regardless of the method, that is the method by which the cell is excited or the intensity of stimulus, the action potential is always the same for any given cell. And this is known as the all or nothing law. And the net height, the action potential is defined as the difference between the potential of the depolarized membrane at the peak of the action potential and the resting potential. And absolute and relative refractory period can be defined as: absolute refractory period is nothing but a brief period of time during which the cell cannot respond to any new stimulus, about 1 millisecond in nerve cell. Okay, and relative refractory period, during another action potential, it can be triggered, but a much stronger stimulation will be required, about several milliseconds in nerve cells. So when a cell is excited, as I said earlier, the action potential, ionic currents begin to flow, and as the action potential travels down the fiber, it cannot re-excite the portion fiber immediately upstream because the refractory period that follows the action potential. Thank you.